Hello students, today I will be dealing with the plasmodesmata. Along with that, I will be dealing with the structure of the cell membrane in detail. So let's start with the plasmodesmata. Now the cell wall, it contains minute pores. Cell wall के अंदर minute pores present है, and what happens through these pores, the cytoplasm of one cell, it is connected with the cytoplasm of the adjacent cell. तो सेल वॉल के अंदर पोर्स प्रेजेंट है और जिसकी वजह से जो एडजेसेंट सेल का साइटोप्लाज्म है वो लिंक होगा. So these cytoplasmic bridges which is formed between the two adjacent cells, these are known as plasmodesmata. So let's write this information. Cytoplasmic bridges between the adjacent cells are called plasmodesmata. So here I will be showing the structure of the plasmodesmata. So ये मैं एक सेल बना रही हूं, प्लांट सेल. प्लांट सेल के अंदर ये जो मैंने अभी स्ट्रक्चर बनाया है, दिस स्ट्रक्चर इज सेल वॉल, एंड इनर टू द सेल वॉल यू नो दैट देयर इज प्रेजेंस ऑफ द प्लाज्मा मेम्ब्रेन. सो दिस इज प्लाज्मा मेम्ब्रेन. Now you can see this structure here. ये जो है ये प्लाज्मोडेस्माटा है और इस प्लाज्मोडेस्माटा की वजह से the cytoplasm of this cell, it is connected with the adjacent cell in the plant. So these cytoplasmic bridges, they are known as plasmodesmata, and you can see that these plasmodesmata, they are surrounded with the help of the cell membrane. इनके आसपास प्लाज्मा मेम्ब्रेन प्रेजेंट है. अब इस प्लाज्मोडेस्माटा के अंदर नजर आता है ट्यूबुलर स्ट्रक्चर. यस, सो आई एम मेकिंग द ट्यूबुलर स्ट्रक्चर हियर. प्लाज्मोडेस्माटा के अंदर नजर आता है ट्यूबुलर स्ट्रक्चर. दिस ट्यूबुलर स्ट्रक्चर इज एक्सटेंशन ऑफ endoplasmic reticulum, and this structure it is known as desmotubule. Yes, तो यहां पर ये जो मैंने ट्यूबुलर स्ट्रक्चर बनाया है, दिस इज डेस्मोटिब्यूल. और ये जो डेस्मोटिब्यूल है, इट इज एक्सटेंशन ऑफ एंडोप्लाज्मिक रेटिकुलम. Actually, this desmotubule, it is connecting the endoplasmic reticulum of one plant cell with the endoplasmic reticulum of other plant cell. So, इस डेस्मोटिब्यूल के थ्रू जो दो एडजेसेंट सेल्स है उनका एंडोप्लाज्मिक रेटिकुलम है वो ज्वाइन हो रहा है. Now, the diameter of pore, the diameter of plasmodesmata ranges from 40 to 50 nanometer. Now the function of the plasmodesmata is that it is involved in the transport exchange of compounds from one cell to the other cell. So it is involved in the transport of mechanicals. And you please keep this in mind that the plasmodesmata, they are present only in the plant cells. Yes? तो ये जो प्लाज्मोडेस्माटा है, ये साइटोप्लाज्मिक कनेक्शन सिर्फ प्लांट सेल्स के अंदर प्रेजेंट है. Now it is very well known to you कि सेल के अंदर जो लिविंग सब्सटेंस प्रेजेंट है, दैट इज प्रोटोप्लाज्म. सेल के अंदर जो लिविंग मटेरियल है, दैट इज प्रोटोप्लाज्म. And with the help of the plasmodesmata, the protoplasm of the one cell is connected with the another cell. तो इसका मतलब प्लाज्मोडेस्माटा क्या कर रहा है? ये लिविंग मटेरियल को ज्वाइन कर रहा है. It is maintaining the continuity of the living material in the plant, and this continuum of living material in the plant, this is known as symplast. So let's write this information. So in the plant, the continuity of living substance, living material is through plasmodesmata. So the continuum of the living material in the plant, this is known as symplast. तो लिविंग मटेरियल के कॉन्टिन्यूम को हम क्या कहेंगे? सिम्प्लास्ट. जब हमने लिविंग मटेरियल का कॉन्टिन्यूम यहां पर देखा है, तो साथ ही हम देख लेते हैं व्हाट इज द कॉन्टिन्यूम ऑफ नॉन लिविंग मटेरियल इन द प्लांट नोन एज. द कॉन्टिन्यूम of non-living material in the plant is formed by cell wall and intercellular spaces. जो नॉन लिविंग मटेरियल का कॉन्टिन्यूम है, दैट इज फॉर्म्ड बाय द सेल वॉल एंड इंटरसेलुलर स्पेसेस, एंड दिस कॉन्टिन्यूम इट इज नोन एज एपोप्लास्ट. इज दिस क्लियर? सो कॉन्टिन्यूम ऑफ लिविंग मटेरियल सिम्प्लास्ट, non-living material apoplast. The terms symplast and apoplast, they were given by the scientist. The name of the scientist is Munch. So after the plasmodesmata, now I will be starting with the cell membrane. So let's start with the cell membrane. Now the cell membrane, it is a thin elastic covering which is present inside as well as outside the cytoplasm. So this is thin, elastic, dynamic, semi-fluid, protective covering which is present inside as well as outside the cytoplasm. Now inside the cytoplasm, you know that the membrane, it is present around the cell organelles. Cytoplasm के अंदर जो membrane है, it is present around the cell organelles, right? And outside the cytoplasm, it forms a boundary. Yes, it forms a boundary outside the cytoplasm. This boundary outside the cytoplasm formed by the membrane, this is known as plasma membrane. तो साइटोप्लाज्म के आसपास बाउंड्री बनाएगी, इसको हम क्या कहेंगे? प्लाज्मा मेम्ब्रेन. So here I will be dealing with the structure of the plasma membrane. So first of all, I will be discussing the components of the plasma membrane. प्लाज्मा मेम्ब्रेन के अंदर जो दो मेन कंपोनेंट्स प्रेजेंट है, दैट इज लिपिड्स एंड प्रोटीन्स, बट अलोंग विद दैट देयर इज आल्सो प्रेजेंस ऑफ कार्बोहाइड्रेट्स, एंजाइम्स एंड वाटर. सो कंपोनेंट्स इंक्लूड्स proteins, lipids, carbohydrates, enzymes, and water. So let's first start with the protein here. The amount of the protein which is associated with the plasma membrane, that ranges from 20 to 70%. Yes. Now these proteins, they can be of several types. On the basis of ease of extraction, they are of two types. Integral proteins, intrinsic proteins. Integral or intrinsic proteins, इनका जो extraction है, that is difficult from the membrane. Yes, the removal from the membrane is difficult. Another one is peripheral or extrinsic proteins, और जो peripheral proteins है, इनका extraction easier है. So we will learn more about the intrinsic and extrinsic proteins. Here just keep this in mind that the extrinsic proteins, they can be easily extracted, but the intrinsic, their extraction is comparatively difficult. Now this was the two categories of the proteins on the basis of the extraction. Now the types of the proteins on the basis of the function they perform. So here the three categories, they can be distinguished. Now the first one is structural proteins. Now the structural proteins, they form the backbone of cell membrane. ये cell membrane की backbone form करेंगी. Now the second one, that is carrier proteins. As the name suggests, these proteins, they are involved in the transport of compounds across the cell membrane. So the function is transport. Next one, that is enzymatic proteins. Enzymatic proteins, they are catalytic in nature. Now after the proteins, let's see what is the condition with respect to the next component, that is lipids. Now the percentage of the lipids associated with the plasma membrane, it can range from 20 to 79%. So this is the range for lipids. So let's write the name of the different types of the lipids associated with the plasma membrane. One is cephalin, then lecithin, cholesterol, along with that there is association of galactolipids. ये जो लिपिड्स का मैंने यहाँ पर नाम लिखा है, इसमें से cephalin and lecithin, they are phospholipids. Both of them, they are phospholipids. For the phospholipids, I am writing PL here. Cholesterol, we have discussed the cholesterol in the earlier chapter also, that this is one of the lipid which provides the stability to the membrane. Now the galactolipids, these are the lipids which are associated with the carbohydrate. Yes, lipid associated with the carbohydrate, that is galactose. So these are galactolipids. और अगर lipid carbohydrate के साथ associated है, they can also be known as glycolipids. Now after this, next component, that is the carbohydrates. The percentage of the carbohydrate associated with the membrane is less as compared to the lipids and proteins. अब देखिए कौन कौन से कार्बोहाइड्रेट प्रेजेंट हो सकते हैं. Hexose sugar can be present. I am writing one example, that is fructose. There can be presence of modified sugar, that is hexosamine. There is presence of sialic acid. Now carbohydrates के बाद, now coming to the enzymes. Now in the plasma membrane there can be presence of about 30 different types of enzyme. तो प्लाज्मा मेम्ब्रेन के अंदर जो कैटालिटिक एक्टिविटी है, that is ample. 30 different types of the enzyme, they can maximally be associated with the plasma membrane. Now the last one, that is water, the percentage is 20. तो ये मैंने यहां पर बताए different components of the plasma membrane. Now here I will be dealing with the structure of the plasma membrane. कैसे इन components का arrangement होगा to form the molecular structure of the plasma membrane. So now here I will be dealing with the different models to explain the molecular structure of plasma membrane. तो प्लाज्मा मेम्ब्रेन के स्ट्रक्चर को एक्सप्लेन करने के लिए आई विल बी डिस्कसिंग हियर 3 मॉडल्स. दैट इज सैंडविच मॉडल, also known as trilamellar model. This model, it was given by the 2 scientists, that is Daniel and Devson. For the second one, that is unit membrane model. Unit membrane model, it was given by the scientist Robertson. Then the next one, which is the most accepted model, that is fluid mosaic model. Fluid mosaic model, it was given by the 2 scientists, that is Singer and Nicholson. So first of all, I will be starting with the Daniel and Devson model, that is the sandwich model. So in the sandwich model, in the trilamellar model, the lipids and proteins, they are present in the distinct layers. So according to the trilamellar model, membrane के अंदर lipids और protein present है and they are present in the distinct layer. So let's see how this distribution, how this arrangement is there. ये यहां पर मैं बना रही हूं phospholipid. फॉस्फोलिपिड के अंदर ये जो पार्ट है, दिस इज नोन एज हेड. दिस हेड इज हाइड्रोफिलिक इन नेचर. और ये जो पार्ट है, these parts they are tails, and these tails they are hydrophobic, and it is known to you that these tails they are made up of fatty acids. So this is one phospholipid. This part of the phospholipid, this is head, and head is water loving, that is hydrophilic. And these are tails. These tails they are made up of the fatty acids, and these tails they are water repelling, or you can say hydrophobic. Now such several phospholipids they will be present in the plasma membrane, and these lipids they are arranged in the form of two layers. प्लाज्मा मेम्ब्रेन के अंदर आप देख रहे हैं जो लिपिड्स प्रेजेंट है, दे आर प्रेजेंट इन द फॉर्म ऑफ द टू लेयर. दिस अरेंजमेंट इट इज अ बाई लेयर अरेंजमेंट. और लिपिड्स का अरेंजमेंट देखो कैसे हो रहा है. जो हाइड्रोफोबिक पार्ट ऑफ लिपिड्स है दैट इज इन द इंटीरियर, एंड द हाइड्रोफिलिक पार्ट दैट इज टुवर्ड्स द आउटर साइड ऑफ द bilayer. And according to this model, the thickness of the lipid bilayer that is 35 Armstrong. अब इस बाई लेयर के ऊपर और बाई लेयर के नीचे प्रेजेंट है प्रोटीन इन द डिस्टिंक्ट लेयर्स. तो बाई लेयर के ऊपर प्रोटीन्स प्रेजेंट है, एंड नीचे प्रोटीन्स present है, and these proteins you can see they are present in the distinct layers. This protein layer it has thickness of 20 Armstrong, this one 20 Armstrong. So you can say that total thickness of the membrane according to this model that comes out to be 75 Armstrong. So 75 Armstrong is the thickness of the membrane. Now according to this model, this membrane is symmetrical. Membrane symmetrical है. Symmetrical का क्या मतलब हुआ? That the two sides of the membrane they are same. The type of the protein present on the two sides of the membrane they are same. Now according to the Daniel and Jeffson model, the types of protein which are associated with the plasma membrane they are alpha globular proteins. Now what are these alpha globular proteins? Let's discuss here. ये मैंने प्रोटीन बनाया. Yes, so you know that protein it consists of amino acid, and these amino acid they are linked with the help of the peptide bond. So this can also be known as polypeptide chain, because the several peptide bonds they are present in the protein. अब ये मैंने स्ट्रेट चेन बनाई है प्रोटीन की. Now what happens? Now the protein it can show coiling. This protein can show coiling forming this helix, and this helix it is stabilized with the help of hydrogen bonds. तो स्ट्रेट चेन ने अब हेलिक्स बनाया, कॉइलिंग शो करी है, एंड दिस हेलिक्स इट इज स्टेबलाइज्ड विद द हेल्प ऑफ द हाइड्रोजन बॉन्ड. सो दिस स्ट्रक्चर विच इज फॉर्म दिस इज नोन एज alpha helix. Now this alpha helical protein again it can show the coiling, yes, and result in the formation of the spherical proteins. Yes, तो देख रहे हैं अब दोबारा यहां पर कॉइलिंग हो रही है एंड दिस फॉर्मेशन ऑफ दी स्फेरिकल प्रोटीन्स. दी स्फेरिकल प्रोटीन्स विच आर फॉर्म्ड हियर, they are known as globular proteins. Right. तो ये मैंने क्या बनाया? Straight chain. Straight chain, this represents primary structure of the protein. फिर प्राइमरी स्ट्रक्चर के बाद ये बना स्ट्रक्चर जहां पर कॉइलिंग हो रही है, हेलिक्स बन रहा है. दिस 2-dimensional structure which is formed, this is secondary structure of protein, and after that you can see there is formation of a 3-dimensional globular structure. This 3-dimensional structure of the protein which is formed here, this is known as tertiary structure. तो यहां पर जो ग्लोबुलर प्रोटीन्स बने हैं, दैट इज द टर्शरी स्ट्रक्चर ऑफ द प्रोटीन. सो दिस इज वेरी मच क्लियर. सो according to this Daniel and Debson model, on the upper side as well as the lower side there is presence of alpha globular protein, and these globular proteins, they are hydrated, that is they are associated with water. So these are hydrated globular proteins. Now let's see what are the features associated with the unit membrane model of Robertson. तो साथ ही में हम यहां पर जो रॉबर्टसन का मॉडल है उसके फीचर्स लिख लेते हैं. Now, according to the unit membrane model, again the membrane is made up of lipids and proteins, and the lipids and proteins, they are present in the distinct layers. And again, the thickness of the unit membrane is 75 Armstrong. 75 Armstrong is the thickness of the membrane according to the unit membrane model. अब ये जो unit membrane model है, according to this, now the membrane is asymmetrical. Why the membrane is asymmetrical? Because now on the two sides of the membrane, the types of proteins which are present, they are different. Yes, the different types of the proteins, they are associated on the two sides of the membrane. Let's see what are these proteins here. Now again I am forming lipid bilayer. Over the lipid bilayer and on the inner side of the lipid bilayer there is presence of proteins, but now these proteins, they are beta proteins. तो यहां पर ऊपर और नीचे now there is formation of presence of beta proteins. तो we can see here slightly how these beta proteins they are formed. ये मैंने बनाया है primary structure of the protein, that is the straight chain is there. Now what happens? Now during the formation of the secondary structures, these protein chains, they can join together with the help of hydrogen bonds. तो सेकेंडरी स्ट्रक्चर जब बनता है तो द प्रोटीन चेन्स दे कैन जॉइन टुगेदर विद द हेल्प ऑफ द हाइड्रोजन फॉर्म्स, दस रिजल्टिंग इन द फॉर्मेशन ऑफ द शीट्स. पर हाइड्रोजन बॉन्ड्स बनने की वजह से ये जो शीट है ये प्लीटेड हो जाएगी, जैसे मैं यहां पर दिखा रही हूं. सो दिस प्रोटीन्स विच आर फॉर्म, दे आर नोन एज बीटा प्लीटेड प्रोटीन्स. राइट. तो पहले केस के अंदर हमने क्या बना, हेलिक्स बनेगा. दीज प्रोटीन्स दे आर नोन एज अल्फा हेलिक्स प्रोटीन. पर यहां पर शीट बनेगी और हाइड्रोजन बॉन्ड बनने की वजह से ये जो शीट है ये प्लीटेड हो जाएगी. तो जब शीट बनेगी तो दीज प्रोटीन्स दे आर नोन एज बीटा प्रोटीन्स. So I am not showing the detail of the beta proteins here. I am showing just the platelet structure will be formed. Now, according to this model, बाहर की तरफ जो proteins present है, they are mucoid, and on the inner side of this membrane, the proteins which are present, they are non-mucoid. So outer side पर mucoid proteins and inner side non-mucoid protein. So what is that mucoid protein? जो प्रोटीन बाहर की तरफ प्रेजेंट है, they are showing the association with the carbohydrate. Yes, they are joined with the carbohydrate. तो जो प्रोटीन्स बाहर की तरफ है, कार्बोहाइड्रेट के साथ एसोसिएशन शो कर रहे हैं, they are mucoid. And on the inner side, the such type of the association is absent. So these are known as non-mucoid protein. So the differences between the trilamellar and the unit membrane model is very much clear to all of you. Now these models, they can also be known as PLLP models. Why PLLP models? क्योंकि आप देख रहे हैं दोनों मॉडल्स के अंदर ये प्रोटीन की लेयर, फिर लिपिड की लेयर, फिर एक और लिपिड की लेयर और फिर प्रोटीन की लेयर. सो दिस प्रेजेंस ऑफ टू प्रोटीन लेयर एंड टू lipid layer, that's why they can also be known as PLLP models. So now these two models, you know, they are the rejected models. So we have to cite certain drawbacks associated with these models. So let's discuss the drawbacks associated with these models. Now one of the drawback is that the thickness of the membrane, it shows the variation. The variation is from 50 to 100 Armstrong. तो अभी हमने यहाँ पर पढ़ा that according to the unit membrane as well as the trilamellar model, the thickness of the membrane is 75 Armstrong. But the thickness of the membrane, it shows variation from 50 to 100 Armstrong. Now the second, these models, they could not explain the movement of polar compounds through the membrane. So now, what do you mean by that? ये मैंने अभी आप बनाया था प्लाज्मा मेम्ब्रेन. प्लाज्मा मेम्ब्रेन के अंदर मैंने दिखाया कि अंदर की तरफ प्रेजेंट है लिपिड बाइलेयर और बाहर की तरफ प्रोटीन प्रेजेंट है. अब लिपिड्स का ये वाला जो पार्ट है, दिस पार्ट इज हाइड्रोफोबिक. यस, ये जो इंटीरियर है, ये जो कोर है, हाइड्रोफोबिक है. अगर कोर हाइड्रोफोबिक है तो इट मीन्स द नॉन पोलर कंपाउंड्स थ्रू द प्लाज्मा मेम्ब्रेन, दे कैन इजिली पास. The core is non-polar, hydrophobic, so the non-polar compounds, they can easily cross through the membrane. But along with the non-polar, there is also transport of the polar compounds. But how the non-polar core, it can cause the transport of the water, polar compounds, that was not explained by these models. So this point is clear. तो जो कोर है वो कैसा है? हाइड्रोफोबिक कोर है. जो हाइड्रोफोबिक कोर है ये वाटर की मूवमेंट कैसे करेगा, पोलर कंपाउंड्स की मूवमेंट कैसे करेगा, दैट वाज नॉट एक्सप्लेन्ड बाय दीज मॉडल्स. नाउ द नेक्स्ट ड्रॉबैक, अकॉर्डिंग टू दीज मॉडल्स, membrane is static. मतलब कि membrane के अंदर जो components present है, lipid है, protein है, they do not have the ability to show the movement. Membrane is static in nature. But we know that the membrane has the ability to show repair. Membrane can show contraction, relaxation, transport, secretion, growth. तो मेम्ब्रेन कितने सारे फंक्शन शो कर सकती है, कितने सारे फीचर शो कर सकती है, कॉन्ट्रैक्शन, रिलैक्सेशन, ग्रोथ, और ये फीचर तभी पॉसिबल हो सकते हैं व्हेन द कंपोनेंट्स ऑफ द मेम्ब्रेन दे हैव द एबिलिटी टू शो मूवमेंट. सो इट मीन्स अकॉर्डिंग टू द प्रेजेंट कंसेप्ट, according to the present models, now the membrane is not static. The membrane, it is dynamic in nature. So these are the different drawbacks associated with these two models. After this, now I will be starting with the most accepted model. That is the fluid mosaic model given by the Singer and Nicholson. So let's start with it.